here today and for presenting our work to us. And as everybody, as we already mentioned, we try to mix a little bit of mathematical and biological talks so that people are not too lost in between them. So people who are mostly in biology will be very comfortable with your presentation, and a little bit less in the mathematical modeling side, but it's the aim is indeed to make a little mixture and then kind of like show people that they can reach out to other people to question in between areas. So Dr. Alejandra Pietro Davó is a researcher at the School of Chemistry in the National Autonomous University of Mexico at CISAL, Yucatan, where she studies sediment-associated bacteria, particularly in cenotes and coastal environments. Her work focuses on exploring microbial diversity and biosynthetic potential to discover new bioactive natural products. And she holds a PhD in oceanography and combines microbial ecology, molecular biology, metagenomics, mainly to investigate these underexplored marine environments. So thank you very much, Alejandra, and the floor is yours.
Thank you so much for inviting me. And as you just mentioned, I'm going to do a 180 here because there's no maths in this talk. But I do remember once participating in a group where we did viral metagenomics, and there were always people from the maths department, and they came up with really good ideas on modeling. So hopefully this talk gives you an idea of what we do in our lab, and maybe you can come up with interesting proposals on mathematical models for exploring the what I call the Maya underground over here. And as you mentioned, I work with sediment bacteria, and I will gladly talk to you about this story. So I know there's no maths, but I'm pretty sure it's going to be fun for you to see at least some of the figures and pictures that I have here. First of all, for those of you that are not familiar with the Yucatan Peninsula, Yucatan is the stretched peninsula that on the southeast of Mexico. It's very close to Cuba and Florida. And this peninsula actually is very famous because if you see here, I really like these two maps because this one shows all of these small dots that you see here are what we call cenotes, and I will talk a little bit about that. And what you can see here, let me use my pointer to make this easier. What you can see here is residue remains of where the asteroid hit 65 million years ago. We're very famous for destroying all life, almost all life on Earth 65 million years ago. This is where the asteroid hit. And as you can see, there is still remains of what that asteroid did in the actual peninsula. So the peninsula was underwater and it emerged about 30 to 35 million years ago. So it's all formed of calcium carbonate, which is what marine microorganisms make as shells. So all of this used to be marine microorganisms, marine organisms, sorry, and they create these shells from calcium carbonate. So all of those deposits of calcium carbonate eventually emerged as this peninsula. So this, our whole area is really part of the biggest underground river in the world because it's like 165,000 kilometers squared, and it encompasses the three states, Yucatan, Quintana Roo, and Campeche in Mexico, and even Belize and Guatemala here. And so I really like this map because it shows all of the little dots are part of these connection to the underground called cenotes. And this map over here shows the fluxes. So this is the highest point in the peninsula is here. And this is basically where all of the flow goes. So it starts here and it goes outside to the ocean, just like a regular river, but it's all underground, right? And the thing here is that every local or every side of this peninsula has its very own local flow, and it changes continuously. It's very dynamic because of its karstic nature, it gets eroded by the water. So it can break a wall and suddenly the flow that goes one side, it erodes and it goes to the other side. So it starts moving a lot. So that changes everything a lot. And one of the things that we explore, as I mentioned, is this these things called cenotes or sinkholes. And what happened here is that the actual platform collapses, and then it creates a huge hole that you can access the river through. And these are incredible places where you can see they're kind of like caves, and then the water is underneath. And this picture doesn't show it very well in these dimensions, but these are actually diverse. So these can be really, really big caves, like flooded with water. And so we decided to, I've always worked with marine sediment bacteria from offshore to inshore. And I work in the School of Chemistry because I work with chemists to look for novel natural products. And then I started with this group of people that studied crustaceans that live in these caves. And they were wondering if there was a way to know what was in the sediments, because that's how they feed. So they were wondering if there was if I could study to tell them what kind of microorganisms were in the caves. And I thought, oh yeah, that's a great idea. And then about 8 years ago, we started studying all of these caves. And then we started noticing, and these are some sketches of the caves. These are two caves. This is a coastal sinkhole called, they call it Hell's Bells in English, Zapote in Spanish, very different names. But what it has is it has these kind of bell-shaped formations that microorganisms help precipitate from all of these minerals and calcium carbonate that is around them. And that's why it has that name. But usually these cenotes or sinkholes have a very big input of organic matter from everything that rains, let's say, from the top. So here it's just a representation of how there's like a dead tree inside. This is very common. There's a lot of trees that fall into them. And then it generates this accumulation of organic matter around it. So we started studying the sediments all around these caves. We divided them in sections. We call it the water mirror, wherever we could get some light. Then the caverns, as you start going into the conduits, because as I showed you in the previous map, like everything over here is interconnected. And then you can get really far into these places and very deep. They can be 65, 70 meters deep. And we have specialized divers that go into these caves, and they can go into the cave system where there's no light whatsoever and very little connection with the outside. So we started noticing trends. This is a paper we published in 2022, and we started noticing that there was definitely a separation between the microbial communities. This is all based on 16S data on microbial communities. And first of all, we noticed that the inland cenote was very different to the coastal cenote, and that all of we'll call it the jungle or the forest, low forest, I really don't know the exact term for it, that's outside the cenote, actually when it comes to microorganisms identity, it clades really well. But then when you start exploring the different sections of those cenotes, there starts to be a trend where you can divide like exactly the sections that we divided. Like the water mirror community is different from the cavern community, which is the triangles, and then the cave, which are the ones that are mostly very well inside all of these conduits, are very different from everything else. So we started exploring that, and we started trying to figure out who were there and what could be their role. So again, this is all based in 16S, but we can identify what kind of microorganisms are there, and then we can propose what kind of metabolisms they have. So what we did find was that the coastal cenotes were very much influenced, of course, because just as the water flows out, seawater flows in, right? So these coastal cenotes are really interesting because they're divided into what they called a meteoric water, which is just fresh water on the top, then a halocline, which is the mix of seawater and fresh water, and then all of the water under it can be marine water, right? So we started noticing that there were there was evidence of marine microorganisms, because they're mostly sulfate reducing bacteria. So these are microorganisms very much involved in the in marine processes, and they are very common in marine water, right? While and some methanogenic archaea also. And while the inland cenote was mostly or had a lot more bacteria associated with the nitrogen cycle. They were ammonia oxidizing bacteria, ammonia nitrate oxidizing bacteria. So they were much more involved, which are all of these over here, with nitrogen pathways, right? And some methyl, because this is methanotrophic bacteria, that were present in the inland. And this all makes sense because, as I mentioned, well, this has influence from the ocean. And there's also, in the inland cenotes, there's a lot of anthropogenic pollution, right? Mostly it comes from the pig farms, because the pig farm industry in the Yucatan Peninsula has grown incredibly incredibly high, and there's like a lot of residues from those pig farms that go on the soil. And then, since it's this karstic environment, the characteristic of this calcium carbonate environment is that it allows everything filtrate into the aquifer. So we're very much affected by all kinds of pollution, because everything is absorbed into the underground river, which is our main water source in the area. So we do have a lot of water, but a lot of that water is very contaminated by human population discharges and by the pig farm discharges. So after we published that, there were a few people that we noticed that a few people had all started working on the water column. All of these that I showed you is from the sediments, and I'll tell you, like, we work on the sediments because the bacteria that we work with for chemistry are mostly and are more dominant in the sediments, not so much in the water column. But then we started looking at things that happen in the water column and that were studied in other groups, and we saw that, for example, this is a very big study on the water column microorganisms in the eastern side of the peninsula and the Caribbean side, and very close to the coast, right? So we started seeing that they saw in the water column a difference in the communities between the meteoric water, the halocline, the saline underground water, because they divided it in 4 sections, and the seawater in the bottom. And this makes total sense, because the bacteria that can tolerate the different conditions and changes in the water column are in themselves different, right? So we started seeing then that there's a pattern that that there's that there actually is in microbial ecology something like they're changing, the communities are changing as the environmental conditions in just a cave change, right? There's also these paper by Brankovits where he suggests that the coastal cenotes, all of these in the either the east or west coast, are actually inverse estuaries. And what it means is that they, as I showed you, and I'm gonna try, sorry if I give you a headache. There we go, this one. As you can see here, like there's a lot of vegetation on top of these cenotes. So all of this is very porous, but there's organic matter degradation over here, right? So the degradation process of organic matter keeps happening as you go down on a land core, because I usually say sediment cores, but this is like dirt core. And if you start going down, you can see how that organic matter is processed until it gets to the production of methane. So what they found was that in the coastal, so I'm going to stay with this one because this is the coastal one. In the coastal environments, what can happen is that there's methane seeping through the walls of the cenote. And that's why it's called an inverse estuary, because usually in an estuary, the organic matter falls and the methane is is processed all the way in the bottom. So you start with you can start with methanogenesis and methanotrophy in the top part of the cenote. And then it can happen again in the bottom part in the sediments, which is also what we found. So now we're seeing all of these like microorganisms that live in this environments, and we're seeing that all of their metabolisms are coupled. I try to generate this with AI. I wasn't very happy, but here it is. But it's how like there's very important metabolisms with respects to carbon fixation, nitrogen processing, nitrogen cycling, so for cycling, and of course, a part of the carbon cycle, the production and consumption of methane, right? So we can see that the sediments actually host anaerobic metabolisms, and they reduce the compounds that are transported, right, or diffused through the redox interfaces. So there's these environments, microbially speaking, are super interesting because you have a water column that the coastal ones mostly, you have a water column where the redox conditions change, and then you have sediments where those redox conditions changed also, right? And it's all coupled. So it's been very interesting to study the microbial ecology of all of these cenotes and all of these environments. And so what we actually do, and this is what not what we actually do, we do both things, but why we started studying these sediments was because of the secondary metabolism. So secondary metabolism is anything that helps a bacterium to defend or to live after it has grown and it's gotten to the stationary phase. So after it's actually done with growing and getting to a population size that is a good size for them to survive, let's say that way, then they start producing chemicals to communicate amongst them, either to say that they're present or to attack other microorganisms and start a chemical warfare. So we start working with those microorganisms, and the way we do this is we go out, and this cenote and like especially cenote polac turn out to be very interesting. We analyzed first a lot of samples just for their polyketide synthase genes to see how they clustered and if they were all the same. And then we saw that this polac cenote was very different from everything else. So we went and explored it a little more. And so it takes like a while, because first, this was in 2021, I believe, when we went. So we were all still pandemic and we went out for about an hour ride south from where we are at station. We're in the Yucatan. We're part of the National University in Mexico, but we're stationed in Yucatan and in a very remote location in Yucatan. That's where the Marine Station is, called Cisal. It's a very small town on the northwestern side of the peninsula. And so you basically you have to get there first to the coastal area, so it's about an hour ride on all of the boats, and then you have to bring equipment in kayaks or these small little what they called alijos here, where you put all of your equipment, because you have to paddle into the cenote, right? And you go through the mangrove, and we have specialized divers that actually can go in these places, because it's very technical diving, and there's not much to see in the dive itself, but the formations and everything under there is really, really cool. And so they go down and they got the samples from the different sediments at different depths, and this is what the samples look like. It was, as I mentioned, 2 different depths. This is what the cenote looks like from the top. So it's in the middle of a mangrove, and you can't really see the connection to anything. So this was a very special place, because we could see that since it's so remote, it was at least less influenced by visitors, right? Because usually all of the cenotes that we go to, you go in your car and then you can go, and now it's very common to go diving in them. So now it's really hard to find places that are not visited by tourism, and this is definitely one of them. It takes forever to get there, and there's nothing let's just say there's nothing amazingly cave speaking to see, but it's very interesting microbiology, right? Microbiologically, I meant. So you have some fresh water in that, a very little fresh water in that cenote. Then you get the brackish water and the halocline, and then you have the water in the bottom, which is very exposed to ocean intrusion, to saline intrusion. So you get all of these different microbes in the water column, but we were interested in the sediments again because we were looking for the secondary metabolites. So we went ahead. Oh, these are the characteristics of the cenote, and as you can see, it's not very fresh water. It starts with a salinity of 20 and it goes all the way to 40. And the reason this happens is that there's not that much. When we visited, it was around I think the 2 times we've been there, it's around September. So it's not so far from rainy season, but it's not so affected by the rain, because even if it rains a lot, it's so hot over here in the tropics that in a day you can evaporate all of that water that rains. So as you can see, it gets almost to 40 in salinity when ocean average is around 33, 34. And then the pH is very it's not bad. It's a very neutral, stable. But and the temperature changes. You see, it's not that much between 26, 27. This is the temperature of the water, 26 and 27 Celsius. So it's always hot here. It's just the degree of hotness that changes. I mean, it's the end of November now, and it's still hot over here, right? But this is what was very important. There is very little dissolved oxygen in the water, and actually you don't see a lot of life in the water column. We did see some fish that were jumping out actually of the water when we arrived, and we think it's fish that get trapped at some point, and then they probably die because there's not enough oxygen in there for them to survive. They're actually jumping, I guess, to try to get even more oxygen or oxygenate the water. And so we explored the microbial composition of these sediments, and we did metagenomes. This is not on 16S, this is on metagenomes, and we started studying what was present in there. We are very interested in the actinomycetes, which is this one, the actinomycetota. The actinomycetes are well known for their production of bioactive secondary metabolites, and that's what we usually look for. And then when we started looking at the metagenomes and trying to find biosynthetic gene clusters, we did find them, but we found, as you can see, in the whole metagenome, we found very few clusters. The depth of 15 meters showed most clusters. There was a total of 10, but still it's very little, right? For a metagenome that has millions of sequences, you really get very little information on secondary metabolism. And the reason this happens usually is because when you sequence, we don't have that great of a coverage of all of the microbial diversity in those sediments, because there's about 10 to the 9 bacteria per gram of sediment. Excuse me. So if you think about it, it's about 5,000 different bacterial species per gram of sediment. That's about the estimate that people have come up with. So when you extract all of the genes from there, you're going to see mainly the genes from primary metabolism, and very few. It's usually between 1 or 2% of your metagenome will actually hit something in secondary metabolism. But we did see some biosynthetic gene clusters. So we decided to explore a little further, and what we did is we did this was done in the lab so that we could we try to reduce that diversity. We know that if you use cultivation methods, and these are different media and different pre-treatments, we increase the temperature in one of them to kill everything that wouldn't survive. And then we also dried the sediments, because this is what we do to actually access actinomycetes mostly. And so we did reduce, if you see, this is like the diversity in the original sample, and this really bright pink here shows you everything else that is not dominant in this sample. So it's really, really diverse. And then we reduced that diversity, and we were able to build some metagenome assembled genomes, because we couldn't, from the original data, we didn't have that much coverage. So you can also get this if you sequence a lot, right? Everything that's in that sediment sample and you sequence it really, like, you get terabytes of information. You can build the genomes of things that are most abundant or yeah, usually the most abundant things that are present. But also, so what we did is that we reduced the diversity, and then we started working, and we started checking, and it did bring us more mags than just doing regular sampling. And in those mags, we did see different types of biosynthetic gene clusters, and it increased a lot. So here's just a figure. We have the paper available if you want to take a look at more closely at the data, but we were able to increase the abundance in the data of these biosynthetic gene clusters. So now we know more or less which kind of bacteria possess these clusters, and we can see what kind of secondary metabolites we can access on these samples, right? So we also did cultivation experiments. We got some actinomycetes, and we basically we sequenced already one of the genomes of one of these streptomycetes, which are one of the dominant genera that possess all of these biosynthetic gene clusters, and that paper is coming up. But yeah, we've seen a lot of interesting things when it comes to secondary metabolism, especially from the cenote. We've seen it in all of the cenotes, but especially from the cenote. So in one of those sampling expeditions around the peninsula, we went to, and I think the map is here, and I hope so. Yeah, here it is. We went to this place in the very close, like in the Caribbean, called Xcalac, and there's a cave here in part of the coastal area. It's an estuary, as you can see here, with groundwater influence just from the bottom, because this is how we get the water from the aquifer. And then there's this cave where some of the divers found these sponges, and so they were very interested in them. And I said, well, bring them over and we'll see what we can grow from them. And it started just as an experiment to see what kind of bacteria were present in the sponges, and what maybe what they were involved in, these bacteria, and if we could get anything that could grow that made any bioactive compounds. So the cultivation experiment is in process. We have found some strains that have genes that belong to the pathways that I showed you. So we want to explore all of the compounds that they make. We're still working on that. But what I'm going to show you here, this is what I meant. This is one of my PhD students' work on 3 different sponges, and he's been taking a look at them. So the sponges come from a cave that's called Bajo de Judas. In this, actually, as you can see, it's a mangrove also, and the cave is about I think it's like 15 or 16 meters depth. And so they go down, they go in the cave, and they started sampling some of the sponges. And here's an interesting thing, is that some of them were on top, on the cave roof, the cave roof, and then some of them were in the sediments in the bottom. And then there was this one sponge that was hanging from, and I think you can see it in the picture actually if I go back. This is the line that the divers put in caves so they can follow and they know how to get in and out, because sometimes this can be very, very hard. It can be very hard to see because there's turbulent fluxes, as I mentioned, that sometimes a current can come in and then you don't see anything. I don't know, they love diving there. I just go to the very open and really like still ones, but they love all of these places. So anyways, they found one of the sponges hanging in there. And the interesting thing is that sponge is closer to sunlight than the others, right? And so we see differences. These are the temperature, the salinity, some of the oxygen data. So as you can see, this one doesn't get anoxic. It gets really low oxic. This is just percent saturation, so not concentration, but it gets to maybe 30 percent saturation of oxygen. So it's not a very well oxygenated cave, but it's not an anoxic cave either. Not like the Polac cenote, which has very, very little oxygen in its water column. I forgot to mention that, but no, I think I did mention that. Anyways, so as you can see, this one has a higher pH, closer to ocean pH, and it's also very hot. It's 36 degrees in there. And so we studied, and this is what my student Adan has been working on. He studied the diversity of the sponges that inhabit these, the microbes that inhabit these sponges. And he has seen differences in each sponge. And this is the one that's hanging from that thread or that line that helps the divers get in and out. And as you can see, I mean, the diversity definitely changes with the different sponges. And these microorganisms are involved in different pathways. Mostly they have genes that are related to carbon metabolism, and he found out that the microbes are using this pathway, the Wood-Lunchal pathway, which actually converts carbon dioxide into acetate, and this is how it fixes the carbon inside in their cells, right? So we see these are the different colors are the different sponges. We see that different sponges have different parts of the pathway. Again, the number of genes involved in each of these pathways is not huge. Because this comes from the metagenomes from the sponges. So a lot of the samples that when you grab the sponge and when you grind the sponge, I was corrected today because I read something and it's not, we all say sponge tissue, but sponges don't have tissue, so sponge cells. So very little, if you compare the amounts, there's a lot of sponge cells and other organisms, higher organisms that live inside the sponge. And then so you get very little coverage of you still get a lot of microorganism data, but in comparison is not that much because this is everything in that sample. So, but we do find genes involved in nitrogen metabolism also, and in nitrogen fixation, nitrogen nitrification and denitrification, right? And all of these are the microorganisms, like these are genes related to microorganisms that live inside these sponges. So it becomes really interesting also in the sulfur metabolism on how all of these microorganisms are helping the sponges adapt to the environment they're in. And one of the very interesting things is that all of these sponges can be actually working towards ameliorating, environmental conditions in that cave, and then that cave water goes out and it feeds into the mangroves around it. So we now, we now want to know about how all of these microorganisms inside the sponges work towards creating the environment that makes those mangroves thrive. And if they're actually doing, like for example, bioremediation of the water that seeps into those caves, because as I mentioned, there's a lot of anthropogenic impact now in all of these areas because their population keeps growing and people keep making more houses near the ocean or more hotels near the ocean, and there is very little control of the waters that come out of all of these populations. There's no water treatment plants in any, like in the Yucatan Peninsula, because and everything just rests right in the, what do they call them? I forgot the name, sorry. It'll come to me, but we don't have water treatment plants. We just have that where you just let the water stand and then it starts flowing out a little cleaner, but it also seeps inside. So we get all of that contamination into the underground river. And you can see that pollution growing because it used to be like whenever you needed to pull water out of the aquifer, you would have to go like 12 meters deep so that you wouldn't get the first part that was more contaminated. And now you have to go all the way to 18 meters deep. So that you can get water that is not contaminated, and it keeps growing. And then there's the saline intrusion that is growing into the underground river because of water extraction, right, for all of the communities. So it's a big problem here in the peninsula that we're starting to address, and it would be very interesting to see how these sponges and their microbiomes and the metabolism from these microbiomes actually fits into all of these, and see if they actually provide any bioremediation. And of course, we keep working with the microbes that we can grow in the lab, their secondary metabolism and the secondary metabolism from the communities inside the sponge. I didn't present the data here because it's not complete, but one of the things my student is doing is the same thing. He's creating MAGs, metagenome assembled genomes from all of these microbial data points from the sponges. And then we're trying to see if we find any secondary metabolism biosynthetic gene clusters, which we have, but we have found very few. As I mentioned before, it's really hard to get coverage when there's so much in there. But and when you don't have so much money to sequence a lot so that you can actually put it together. So we usually put together what's most abundant. Okay, so that's what I wanted to tell you about what we do in the lab. Thank you so much. And as I always say, I mean, this is not a one-person job. It will, as you very well may know, this is all possible because everybody gathers, and there's a lot of students, a lot of techs, and a lot of people involved. And I want to thank each and every one of them. And of course, all of the funding opportunities that we've had. It's been actually a lot of fun to explore all of these caves. And if you want to see more pictures and videos, please let me know, and I'll try to fish them from my computer if you want to see them right now. But before I leave, I want to make a commercial. Anybody here that's interested in marine natural products, we have this the International Symposium on Marine Natural Products coming up in next October, and it's going to be held and happening in the Riviera Maya. It's a really great place to come and visit. It's right there in the Caribbean, and this is an all-inclusive hotel that we're going to have it in, so it's going to be a lot of fun. So I'm going to leave this here so that you guys can either, I'll actually try to copy it in the chat in case you're interested, and I will take any questions.